Hi everyone. So today under the cytoskeleton we are going to learn about actin filaments, which is one of the three components of cytoskeleton. And you already learned about intermediate filaments and microtubules. So in today's lecture, let's discuss a little bit about actin filaments. So actin filaments can be found in many eukaryotic cells. These are actually very abundant, and they are actually involved in many cells movements. So without actin filaments, animal cells may not be able to move along the surfaces. So similar to microtubules, actin filaments are unstable, but they also can become very stable by associating with other proteins, and these proteins are called actin binding proteins. So depending on which actin binding proteins that these filaments and monomers associate with, they can make very durable stable structures in cells. So for an example, you can find actin filaments making these microvilli that are found in gut epithelial cells that increase the surface ratio in the gut surface area of the gut cells that improve the absorption. And then they can make these small contractile bundles that can be found in the cytoplasm, and they also can make these stable structures like finger-like dynamic phylopodias in the edge of moving cells. And then when the cells dividing, they also can help cell division, cytokinesis. So when you think about actin filaments, if you look under the electron microscope, you can find that actin filaments are much thinner and much more flexible compared to microtubules. So they're about 7 nanometer in diameter, and these are actually, each filament is actually a twisted chain of identical globular actin monomers. So actin is actually made out of these subunit called actin monomer. So these monomers can come together, bind, and then they will make actin filaments. So monomers are also called globular actin or G-actin. And when they come and make these filamentous actin is called F-actin. So these actin filaments can be found in many structures, like the one that we just talked in the previous slide. So like microtubules, these actin filaments also have a structural polarity that has a positive end and a negative end. We also call the positive end as the barbered end and the minus end as the pointed end. And so they are actually very abundant, like I mentioned, in the cells, and then they make these stable structures. Now let's look at a little bit about how actin polymerization can happen. So actin filaments can grow by adding actin monomers to either end. So actin monomers, on the other hand, carry this bound ATP. So when actin monomers are added to either end, this bounded ATP is hydrolyzed upon adding to the filament. So when the ATP is hydrolyzed, the stability of the polymer reduces. So like I mentioned, actin monomers can bind from both the plus and the minus end, but growth can be faster in the plus end compared to minus end. This is because upon binding, these globular actin bounded ATP can become converted or hydrolyzed into ADP. And when ATP is hydrolyzed to ADP, it reduces the stability of that monomer binding to the keep bound to the filament. So these ADP bound monomers will remain in the actin filament until a TP bound monomer is added to the plus end. So then these ADP bound monomers will release from the minus end. Something very important to remember is that what is the concentration of free actin monomers in the cytoplasm. Depending on that, the polymerization of filamentous actin can vary. So for an example, if the concentration of free actin monomer is high in the cytoplasm, actin filaments can grow from both ends, both the plus end and the minus end. So it will rapidly grow into filamentous actin. But if there is an intermediate concentration of actin monomers in the cytoplasm, you will see this scenario called thread milling. So thread milling happens when in the intermediate concentration of actin filaments, the plus end actually grows faster than the minus end. This is because the actin monomers will added, the ATP bound actin monomers will added faster to the plus end before it is hydrolyzed. So positive end or plus end will actually grow faster, and in compared to the plus end, in the minus end, the ATP is hydrolyzed faster than new monomers can be added. So therefore, the filament loses the subunits from the minus end while subunits are added to the plus end. So this will actually look like thread milling, as you can see in the picture. So when the rates of addition and losses are equal, the filament can remain in the same size. So cells can determine if the actin filaments need to be growing from the plus end or minus end. Also, if the cell need to keep the actin length, filamentous actin length in the same. So all they got to do is increase or decrease the free actin monomers in the cytoplasm. So most actin in animal cells can assemble into actin filaments, while the other half remain as the free actin monomers. So like we saw in the previous slide, so when you control the free actin monomers, you can control the behavior of actin. So how do cells actually control actin, free actin monomers from polymerizing? So cells actually utilize these different kinds of proteins to control the behavior of actin monomers. So for an example, cells can actually utilize these small actin binding proteins called thymosin and profilin. When they bind to these free actin monomers in the cytosol, they will prevent actin monomers from polymerizing. But when the cell actually require um, filamentous actin, so they can use another protein called formin to promote actin polymerization. So far we have found large number of actin binding proteins in cells that can control and assemble actin. So let's look at several of functions these actin binding proteins can provide in cells. So one, they can hold actin filaments together in parallel bundles in microvilli like we saw earlier. They can cross-link actin filaments in gel-like meshwork in cell cortex, like in red blood cells. They can actually use these several of these proteins to sever actin, so cut actin filaments into shorter fragments. And also they can associate these myosin motor proteins to form contractile bundles. So this picture actually show all different kinds of functions these actin binding proteins can provide to cells. So in the first here you can see actin monomers. So actin monomers are found in the cytosol. So you have to remember these are globular actin monomers. They have bounded ATP. So sometimes cells can use monomer sequestering protein that will prevent them from going into filaments. Sometimes cells can use these nucleating proteins such as formin. They will actually look like a cap here in the front here. So these are nucleating proteins. And then cells can use severing proteins. Severing proteins actually cut filamentous actin into small fragments. Cells also use these cross-linking proteins in the cell cortex, which we'll talk about in a little bit, to keep actin in a meshwork in underneath of the plasma membrane. And when cells use capping protein, they will block the plus end of the actin filaments. So polymerization from the plus end is further blocked. And then they can use side binding proteins like tropomycins. This is also bound the actin filaments. And then they will use myosin motor proteins and bundling proteins in filopodia. So these are all different kinds of functions actin binding proteins can provide to cells. So far we talked a lot about cell cortex. So what is cell cortex? So you learned previously that cell cortex is actually just beneath the plasma membrane. You can find this meshwork that provides support to the plasma membrane. So plasma membrane is thin and fragile. So in order to provide mechanical strength to plasma membrane, we have this highly concentrated actin layer just beneath the plasma membrane. So these actin filaments are linked by actin binding protein into meshwork that actually support the plasma membrane. So since animal cells do not have a cell wall, this cell cortex actually provide mechanical strength to these plasma membrane. So previously we talked about red blood cells and the actin cell cortex that you can find. So this cell cortex actually provide mechanical strength. This picture also shows the cortex that's made out of actin filament. In red blood cells, you can actually find spectrin filaments as well, which provide that mechanical strength to red blood cells. One of the another functions that actin filaments provide to cell is allowing cells to crawl. So many eukaryotic cells actually move by pulling themselves across surfaces. This is not similar to swimming motions. So white blood cells, they can move from blood to infected tissues when they recognize the small molecules released by pathogens. So in order for them to move, the actin filaments in the cytosol help these movement or cell crawling. So the chemotactic molecules bind to receptors on cell surface that trigger the changes in actin filament assembly that allow cells to crawl. So there are at least 3 interrelated processes that helps to steer the cells toward their target. So these are actually, first one is a cell send out protrusions at the front of the leading edge. So these protrusions actually have filamentous actin where their plus end is facing the plasma membrane. So actin can polymerize towards the plus end, and that will help to make protrusion in the leading edge. Second, these protrusions adhere to the surface over which cell is crawling. So they use these different kinds of molecules on the surfaces of cells to bind to the cell surfaces. So when they bound to the cell surface, the third part where the rest of the cell can drag itself toward the traction on this point of anchoring. This way, the cell can actually crawl. So all 3 of these processes involve actin in different ways. So cell crawling is actually supported by the actin cytoskeleton. So in summary, actin filaments are actually helical polymers of globular actin monomers. These actin filaments are also polarized compared to tubulin. They also have fast-growing plus N and a slow-growing minus N. So assembly and disassembly of actin filaments are controlled by hydrolysis of ATP bound to actin monomers. In cells, actin filaments are controlled by different actin binding proteins. Cell cortex is actually just beneath the plasma membrane. You can find this concentrated network of actin filaments that provide mechanical strength to cells. And we did also talk about cell crawling, which is supported by actin filaments. Cell crawling is important. It is non-swimming motion of cells that is needed to move towards a target. So I hope this helped. Thank you very much.